Welcome, everyone. Thank you for attending our third virtual Oskar Fischer lecture, which is the fifth lecture in a series that was launched in the spring of 2020. I'd like to acknowledge the collaboration and representation of many other schools and organizations across the UT campus and also nationally for joining us today. And we are particularly indebted to Dr. Jim Truchard and his vision for keeping Oskar Fischer and his research alive in perpetuity through this lecture series. Today's speaker is Marc Diamond from UT Southwestern. And following Marc's presentation, Drs. Bill Seeley from UCSF and Jeffrey Kelly from Scripps Research in California will join us as our expert discussants. There will also be time afterwards to answer questions, so please submit questions at any time throughout the lecture, using the Ask Question button on your screen. And now I'd like to introduce our speaker for today. Marc Diamond and we've known each other since Marc was in high school, in full disclosure, and I met him when I was a resident at UCSF and then as a postdoc in the neurology department there. He went to Princeton for his undergraduate studies, and he graduated with a degree in history in 1987. We first worked together in the laboratory during the summer of 1984, and I'm proud to say that I taught him how to make his very first buffer, laying the foundation for everything he has done since. But all kidding aside, we developed a very good friendship at the time, and that's continued to the present. After graduation from Princeton, Marc entered the UCSF School of Medicine, and he had a life-changing research experience in Keith Yamamoto's lab, where he studied steroid receptor control of gene expression. And that led to his very first paper, in which he was first author, and it happened to be in Science in 1990. After finishing med school at UCSF, he did a residency in neurology and became chief resident there in his final year. Then he did a postdoctoral fellowship with Keith Yamamoto once again, and then became assistant professor of neurology at UCSF in 2002, moving up the ranks to associate professor in 2009. That same year, he was then recruited to Washington University, where he became a full professor in 2013. Then in 2014, was recruited to UT Southwestern as founding director of the Center for Alzheimer's and Neurodegenerative Diseases, and as a professor of neurology and neuroscience. Marc's research focuses on molecular mechanisms of neurodegeneration with the goal of developing novel therapies and diagnostic tools to detect and stop neurodegeneration before symptoms present. In that vein, a therapeutic antibody he co-developed at Washington University in St. Louis is now entering clinical trials for the treatment of dementia. He will speak today about his intriguing work on the biology of the protein tau, which is central to several neurodegenerative disorders known collectively as the tauopathies. Marc's research has revealed how tau forms disease-specific structures that seed and self-propagate across the brain. Marc, we are honored to have you here today. Thank you for joining us.
Thank you very much, Bob. I'm really glad you didn't go too far into my early days with you. I seem to remember a lot of naps. Anyway, I am really honored to give this lecture, and I'm also very pleased to learn more about Oskar Fischer, which I did with Dr. Truchard this morning, and as well because of reading about Dr. Fischer. Today, the title of my talk is Structural Classification of Tauopathies, which sounds very sort of dialed in and hyper-specific, but actually the general ideas behind this talk really speak to a much more general phenomenon about prion mechanisms in neurodegenerative diseases. And I hope all of this will begin to make sense by the end of the talk. I need to disclose a few things before I go too far. The first is that, as Bob mentioned, when I was at Washington University, I developed a few things that have forms of IP. One was some antibodies that were licensed by C2N and are in clinical trials, but also biosensor cell lines are widely used by industry and academic labs. And these represent conflicts of interest, but don't really have anything to materially to relate to what I'm talking about today. I wanted to start by thanking the members of my lab who moved with me from Washington University to Texas. This is when shortly after we arrived, and I'm actually going to be referring to work by a number of these people, especially in the beginning, Brandon Holmes here, who's now a senior resident at UCSF, Sarah Kaufman, who's in her second, just about to start her R4 year as well at UCSF, David Sanders, who's a postdoc at Cliff Brangwine's lab at Princeton, and Hilda Merbaha is a neuropathology or a pathology resident at UT Southwestern right now. But I want to emphasize especially Jaime Vaquer Elisea, whose work is going to feature prominently at the end of this talk, and he's an enormously talented graduate student currently in the lab. So most of us have heard something about prions. They were discovered at UCSF by Stan Prusner, and for which he was given the Nobel Prize. And I think a fundamental idea about prions, which are infectious proteins, is that they achieve this pathological function by undergoing a fundamental change in their shape, in their three-dimensional conformation. And this is a cartoon of putative structure of the prion protein in its normal form, which is rich in alpha helix, and when it converts to a pathological form, which Stan Prusner referred to as the scrapey form, hence PRPSC, which has a lot of beta sheet structure and is capable of forming pathological assemblies that self-replicate in living systems. One of the interesting features about prion diseases is that there are many of them, and each of them derives from unique protein conformations or shapes in which this pathological form can assemble. And we call those different shapes strains. Now, what I'm going to be emphasizing for this talk is that the rules of prions apply in virtually every respect to the tau protein. And hence, we can learn a lot about the biology of tau as it causes diseases based on principles derived originally from Stan Prusner's work. The tau protein was originally identified by Don Cleveland when he was in Mark Kirshner's lab in the mid-70s at UCSF. And it was identified because it binds tubulin in the cells. And this is an image below showing a tau, full-length tau protein fused to a fluorescent tag and a stain for tubulin in red. And you can see in orange that the two are perfectly co-localized in a cultured cell. But about 10 years after its initial discovery, tau was discovered to be the component of neurofibrillary tangles in Alzheimer's disease. And about 10 years after that, a series of familial mutations were identified in the protein, which cluster in this domain we call the repeat domain, which cause dominantly inherited neurodegenerative disease that is utterly distinct from Alzheimer's disease. So it was the identification of these point mutants that really solidified the understanding that the tau protein is itself a cause of neurodegenerative diseases. Now, the way we have come to think about it in my lab is that tau is really the final common trigger for neurodegeneration from a variety of causes. This can include genetics, as I've mentioned, the protein amyloid beta, which leads to tauopathy, which we know as Alzheimer's disease. As well, we know that various inflammatory states of the brain can lead to progressive tau deposition, brain trauma, and just getting old. And all of these different triggers lead to distinct types of tauopathies. There's probably over 30 different ones that can be identified, and we classify them as tauopathies because they have tau accumulated in ordered assemblies in the brain. And this is just a picture of neurofibrillary tangles from an Alzheimer brain. Now, when I was very early on in my training, I actually saw a patient with a disease called primary progressive aphasia in the clinic. And I had no idea that this was a tauopathy at the time. And what struck me was the fact that a patient could have very isolated degeneration of one part of the brain. And I don't have that patient's scan. These are pictures given to me by Bill Seeley a number of years ago. And it shows the exquisite regional specificity of distinct tauopathies, where much of the brain can be fully intact, but just small subregions degenerate. In this case, the temporal parietal region, or the temporal lobes, or the frontal lobe. And of course, as distinct regions of brain degenerate, you get distinct clinical syndromes, of which there are many. The key take-home here, though, is that this one protein can cause multiple diseases. And as I was growing aware of this during my training, the other thing that became clear was that brain networks appeared to be linked to neurodegenerative diseases. And knowing what I did about prion disease, I came up with a model about 20 years ago, although it took some time to get the work published, for how tau might work as a prion. And the idea is pretty simple, that you have a protein in one state, a healthy state in a neuron, but it can somehow convert to a pathological form that would form these ordered assemblies, which, if they happen to be mobile and could transmit pathology from the outside to the inside of a cell, could then propagate disease through brain networks. And this could simultaneously explain the diversity of different tauopathies, the different brain networks that are involved by them, and also the common features in terms of amyloid or ordered assembly formation that the tau protein had with the prion protein. So when I was an important aspect of the prion protein is the fact that it really serves as a template for its own replication. And what we mean by this is that a protein can go from one conformational state to another, as represented here by an oval to a rectangle, and then those rectangles can assemble, but they can assemble in different shapes. The one on the bottom here would be a helix. The one on the top would be a straight filament. These are obviously cartoons. But the idea here, though, is that through a remarkable property that we don't fully understand, the ordered assembly serves as a template for its own replication inside a cell, such that native protein is added on to a growing assembly, and depending on the shape of the template, you get a different fibril out. And this process of faithful replication of unique structures in living systems gives rise to what we call strains. And the important idea here from the prion field is that if you know the structure, you can predict the type of pathology, you can predict the syndrome, because the structure is itself causal of the disease. And we think the exact same principles apply to the tau protein. In work I really don't have time to describe, in the last 5 or 6 years or so, we have developed the idea that this pathological form of the tau protein is in fact, in its first instance, a single monomer, which can adopt multiple distinct structures, which themselves can build different types of assemblies, as again illustrated in these cartoons. And I would love to come back and tell you a lot more about the nitty-gritty structural biology of this process. Suffice it to say that all this work started back at UCSF when Bess Frost was a graduate student in my lab, and she figured out a way to use existing protocols to make tau fibrils from recombinant protein. She wanted to test the idea that tau could be a prion, and the simplest way we thought we could do this was to culture cells in which we had fused tau to the yellow fluorescent protein, like I originally showed you. Bess added these fibrils to the cell media, and in a small fraction of the cells, she saw that the tau protein that had been expressed inside the cell converted from its normal form to an aggregated form that actually had an amyloid structure and will stain with thioflavin. So this was a remarkable finding for us. It told us that the prion hypothesis could be true. We needed to develop better systems to study this, and so we developed the biosensor cells that I have previously referred to. These involve taking the tau protein and developing a fragment of it called the repeat domain, which we fuse to complementary fluorescent protein tags. In the first instance, we use yellow and cyan fluorescent proteins. These are expressed inside a cell and remain as stable monomers. They are non-pathological until the cell is exposed to a tau seed or ordered assembly. And that could be a single monomer in the right conformation or a larger assembly indicated here by these triangles. When these get into the cell, they trigger a catastrophic formation of intracellular fibrils or amyloids or aggregates, however you want to refer to them. And those bring the fluorescent proteins into close proximity, and you get a FRET signal that we can measure in many different ways. The simplest way has been to use flow cytometry to measure individual cells. And using this, we created a highly sensitive and specific assay with an enormous dynamic range of roughly 2 to 3 logs of sensitivity of linear range, as well as dynamic range. And this is our first generation of biosensors, which were sensitive down into the hundreds of femtomolar of tau monomer equivalents. We have now a second generation, which is approximately 100 times to 1,000 times more sensitive, and we're into the range of attomolar detection. These biosensor cells have been now widely used in industry, and many people find them useful to detect pathological forms of tau. When we had these cell lines going in the lab, my student David Sanders began to wonder about the idea of strains. We were still exploring. We wanted to test whether tau could do what the prion protein does. That is to say, form stably propagating structures that are different from one another and are actually transmissible. And he created tau fibrils in vitro and seeded them by exposing these stable cell lines that express tau fused to YFP. And he made a remarkable observation that a preparation of fibrils added to these stable cells, which only have tau monomer, led to a variety of distinct phenotypes in the cells that had been successfully seeded. I'm just showing this in cartoon form here, but he used these just by visualization under a microscope to clone out individual lines of cells that perpetually maintained tau in an aggregated state. And this is an actual picture of 2 of the more than 30 different strains we have now created in the lab. The one is called DS9, the second DS10. You can immediately appreciate the different patterns inside the cell that the tau takes as it begins to aggregate. And what we have subsequently done, and I don't have time to tell you about, is link that these different structures inside the cell to different shapes of tau. What's very interesting is that these strains are transmissible. David showed that he could take an extract of this cell line, and by exposing a naive cell line to this structured of tau, he could recreate the same cell line. This was true for all the clones he identified. And what was amazing to me was that these were indefinitely perpetuated. In other words, once the tau had formed this pathological structure, the cell would stably maintain it as it divided. This gave us an effectively infinite supply of tau in distinct conformations, something no other lab had. And David and his colleague, Sarah Kaufman, who was an MSTP student, began an exhaustive study of these strains in animals. We used cell lysates to test whether tau could be an infectious protein. We did this using the same basic methods that Stan Prusner had pioneered in the preceding decades. In this case, however, rather than a brain lysate, we took a cell lysate from cells that were derived originally from our recombinant tau seeds. We inoculated the brains of a mouse that carried a human tau gene with a disease-associated mutation that poised it, making it ready to form pathological aggregates. Sarah inoculated the brains of these mice in the hippocampus. She waited 4 weeks, a very short period, sacrificed the animal, and observed that we saw robust induction of tau pathology. This had previously been observed about 5 years before by the lab of Marcus Tolnay. But what Sarah and David were able to do was to purify the human tau back out of this mouse, inoculate a second and a third generation of mice, in each case propagating an identical pathological pattern that related back to the original strain used. Finally, they took the tau from this third generation of mice and inoculated naive cells, and they were able to recreate the original cell lines. So this is just a picture of DS9 cells passaged 3 times through living animals with a completely different form of tau, and then the cells re-inoculated created the same structure. The same was true for this clone 10. At this point, we started talking about tau as a prion. We had created pathological forms in a test tube, inoculated cells, created cell lines, and created a unique forms of neuropathology in the mice that was utterly transmissible between animals. However, Sarah and David wanted to take this further, and they did an exhaustive study of 18 distinct tau clones that we had propagated in cells. In each case, inoculating the mouse and then tracking the propagation patterns of the pathology over time in the animals. And what they observed was that each strain had its unique patterns of propagation, timelines, and patterns of cell pathology. I'm just showing 2 of the 18 examples here. One clone gave a very sort of staining of dendritic arbors and filled cell bodies. Another stains axons and is much more robust at propagation. The point here is that we at this point knew that tau could form distinct structures that would create unique pathologies. The next step for us was to go into human brain and ask what were the strains that we could isolate from a variety of tauopathies. And to do this, we had to collaborate with a talented neuropathologist. And we found in Bill Seeley, both a neuropathologist and a neurologist. We took brains that Bill had characterized and inoculated our cell lines. And David isolated hundreds of individual clones and characterized them by their morphologies. You can see in these different pictures the different ways that tau could look. These were immediately identifiable even to an undergraduate or a postgraduate student in the lab whom David taught to recognize the patterns. He then asked her in a blinded fashion just to characterize all the clones that she could see, and she looked at hundreds. And then we went back and grouped the clones according to the patients they come from. We saw, and here's the results. Basically, the color codes refer to the different strains or types of tau structures we were identifying. The numbers here refer to individual patients. And what you can immediately see is that Alzheimer's patients are dominated by one type of tau strain, and everybody looks the same pretty much. But if you look at a different tauopathy, a group of patients classified by Bill as, for example, having progressive supranuclear palsy, we could see that rather than one dominant form, we saw multiple subtypes. This told us that within the neuropathological diagnosis, we were probably missing subsets of patients with different types of tau strains. But at this point, we felt very confident in asserting, again, that tau really had all the essential features of a prion. We could take individual structures, inoculate a naive mouse, and create new patterns of neuropathology that were transmissible. At the same time, we could take humans and isolate distinct strains that correlated with their underlying neuropathological diagnosis. So this body of work really began, for me at least, to put tau assemblies at the core of understanding how tauopathies evolve. Now, at the same time, as we were developing these methods, labs focused, especially in Cambridge, in the lab of George Sherris, in collaboration with Michel Godert, had really advanced the technology of cryoelectron microscopy to physically study fibrils in a way that no one had been able to do before. This is a very labor-intensive method that involves biochemical extraction of brain to pull out the detergent, insoluble forms of the protein of interest, in this case, tau. These are placed under specialized electron microscopy grids, imaged, and thousands of images are obtained. The investigator must then pick the images and use computer software to reconstruct a more detailed atomic image of the fibrils. In this case, they were able to solve the structure of the fibrils that occur in Alzheimer's and a range of tauopathies, and have really transformed our knowledge at an atomic level of these fibrils. But there are some limitations to this method. First, it's incredibly labor intensive. It takes months, and many labs don't have the capacity to do this. The second thing is that it's actually fairly biased because it involves a very rigorous biochemical extraction of brain. It involves basically cherry picking images for analysis and ignoring images that cannot be analyzed. So despite its biases, it's immensely powerful, and we should all be excited about it. Using this method, predominantly the Sherris lab has solved the structures from fibrils derived from many different tauopathies. And these are just images taken from a recent paper from that lab. But they involve, you can appreciate at a relatively quick level that these folds of the tau protein, which are basically a view of a fibril seen from above and represent the folds of tau monomer stacked on one another, really differ between diseases. I'll draw your attention to the Alzheimer's structures, which represent two different types of filament structures, a paired helical or a straight filament. And you can see the underlying monomer structure is the same, but it's just oriented either around this hook here or more towards the back end of the hook. Now, we are not cryoelectron microscopy experts in my lab, and I was very interested in the idea that we might be able to deconvolve the complex structures of tau using simpler means that might be translated more broadly. And our goals were several-fold. First, we wanted to try to figure out a way to rapidly sort out tauopathy brains using relatively small amounts of brain, not the grams of material required for cryoelectron microscopy. Secondly, we were really interested in testing the fidelity of the cell system to accurately replicate prion phenomena for tau. And finally, we wanted our system to be as unbiased as possible. And to do this, I got the idea of the species barrier. So species barriers exist in prion disease, and that's why deer have prion disease that's generally not easily transmissible to sheep. The sheep disease cannot be transmissible to humans, but it can be transmitted to a cow, and a cow can occasionally transmit a prion disease to a human who has the right mutations. But one of the principles of the species barrier is that the reason prion diseases tend to stay within the animal that they develop in is that even individual mutations in the prion protein can block its ability to act as a template, such that a seed from one animal that might come into contact with a very similar prion protein with a point mutant is unable to serve as a template because of those amino acid changes. We exploited this idea. Rather than waiting for mutations, we engineered mutations in the form of alanine substitutions throughout the repeat domain of the tau protein. And we engineered them in an arrayed form. And a way to think about this is if you imagine the tau protein as a Lego block with nubbins and holes, we are effectively either filling a hole or sawing off a nubbin, and thereby changing the way that the blocks might assemble. The idea was that if you had a cell that had tau of a certain assembly conformation, and you expose it to a tau monomer, if the alanine substitution is in a position that would interfere with its incorporation into the assembly, for example, into the circles here, you would get no incorporation. And because the tau inside the cell has a YFP tag on it, there would be no fret signal. On the other hand, if the cell had an aggregate that was compatible with this mutation, you would get a fret signal. So we're effectively testing for, in a positional way, we're sampling through the tau molecule, asking what amino acids are required to build any particular structure. And my student Jaime developed a system to amplify the structures from either the clones or human brain and then test them. And I'm going to show you the results now. The first thing to do was to test the reproducibility of this system. How reliable was it? So what we're seeing here is a graph that shows the effect of individual mutations mapped onto the repeat domain of tau, which I'm just cartooning below here. And every time you see a dip in a curve, that means there's a mutation at a particular site that was very disruptive for incorporation of that particular strain. In this case, we're looking at a strain called DS16, and we just did 2 different replicates of this in which we tested a library of tau mutants and asked how each of about 150 mutants could be incorporated into the strain. And what you see is there's some hotspots. These are places that are really critical for the assembly. You can see right at the junction of the second and third repeat domain, there's a spot within the third repeat. Another way to look at the data is just to correlate each amino acid substitution by its across replicates, and there's a very, very high correlation rate. These are basically telling us the same thing. But with the correlation of the testing of each amino acid, you then get a simple number that tells you how similar 2 strains are to one another, something that would be very, very difficult to do if you were simply looking at the structures produced by cryoelectron microscopy, for example. So this is an example of a study of 2 strains that are highly dissimilar, strain 8 and strain 7. You can see there's very poor correlation by amino acid. And if you look at the map of where these mutations fall, you can see that the strain number 7 relies on the 3rd and 4th repeats of tau in this core domain. Hits here affect strain 7, but have no effect on strain 8. Whereas mutations in the 2nd repeat and a little bit in the 3rd here affect the strain 8, but not strain 7. By contrast, here are 2 strains which we think actually may be identical. They were, even though they were isolated separately, and you can see the curves are almost overlapping. The R-squared values are very close to 1. Jaime then took every strain and compared it to every other strain and clustered them based on their similarity. And what I've done here is create a map where basically the the colored boxes highlight groups of strains that were judged to be not identical, but very, very similar based on this alanine scan method. You can see 8, 4, and 17 are very similar to one another, 2, 10, 19, and 3, and so forth. So this is a way of, in an unbiased fashion, to compare the relatedness of distinct types of tau assemblies. And what was amazing to us is we took this data and went back to the vast trove of neuropathological data derived from animal studies. And we found that, in fact, the strains that were similar based on their amino acid requirements to assemble created very, very similar patterns of neuropathology. For example, 8, 4, and 17, you can immediately appreciate the cell body and axonal patterns as compared to, say, strains 18 and 7. So what this tells us is, in a very refined way, that we can, going way beyond cell morphology, that we can map out and essentially fingerprint a tau strain and identify it without going through a labor-intensive extraction and cryoelectron microscopy protocol. We have now used this method looking at the amino acid requirements of different tauopathy brains. So in this case, we're comparing 2 Alzheimer's brains from different patients. And you can appreciate that they have essentially overlapping curves, and they require the same amino acids to build their structures. What fell out of this analysis, however, is a fascinating realization that the cryoelectron microscopy cores identified in prior studies did not do justice to the entirety of the tau protein. And in this case, I've drawn a box around valine at position 306, which is the last amino acid visualized in the cryoelectron microscopy image as being part of the Alzheimer's core. But yet we can see there are amino acids N-terminal to this that are not visualized in the core, yet still play a really critical role in the stability of the tau assembly. This is, by contrast, comparing an Alzheimer brain versus a CBD brain. And what you can appreciate is that the CBD brain, which is in green, makes no use of, or very little use of, the amino acids at the C-terminal portion of the repeat domain, whereas the Alzheimer brain requires these amino acids in order to build a stable fibril. Earlier, more proximal in the repeat domain, however, the 2 different structures actually use a similar core. So you can begin to appreciate, with this fingerprinting method, we're actually asking sort of what we think of as energetic questions about the role of distinct amino acids in building these pathological structures. Which brings us close to the end of the talk, and I want to just come back to the cryoelectron microscopy image of a tau fibril from an Alzheimer brain. And in this published image, the authors from the Sherris group highlighted densities in the structure that could not be identified in the cryoelectron microscopy method because they are mobile. They're present, but they're not holding still, and therefore you can't get a good image of them. The core, which begins at that valine 306, is stable and is visualizable. Remember that these amino acids, that with if we imagine the protein forming a loop here, this could represent amino acids that we found to be required for forming the fibrils. So that led us to collaborate with the lab of Łukasz Joachimiak, one of my center faculty, and a talented post-baccalaureate student in his lab, Vish Malapudi. And what Vish did is use a software that individually basically took the structure of a cryoelectron microscopy fibril and used computational approaches to ask in an iterative way about each individual amino acid in the structure and its relative contribution to the energetics of that structure if it was to be stacked. It's effectively using the computer to simulate what would happen if you made a mutation at any one of the structures and then ask about the energetic stability, the consequences of that mutation. And the in silico map gives you in red the hotspots, that is to say, the amino acids in the Alzheimer filament that are likely to be playing a really critical role in its energetics, in its stability. On the right-hand side, and very closely matching, you can appreciate the results of the alanine scan. Now, basically what this is telling us is that the alanine scan in this case was reporting on amino acid without any prior knowledge of the structure. We were getting hits on amino acids that would have been predicted based on a published fibril structure. Now, it doesn't hold true for the entire molecule, and you might ask why. And we believe that that is because the energetic predictions of Rosetta can only use atoms that it has structural information or coordinates for, whereas our study actually just uses an unbiased genetic approach. So we get hits in this parallel region that do not match. And we believe that is because of that loop that I mentioned earlier of domains that are present and part of the structure, but are not holding still enough, basically. So the point being overall, though, that our cell models in many respects match the structures that we are detecting in patients. This gives us a lot of excitement because it suggests that we might be able to replicate the structures specifically from the patients in the cell models at an atomic with precision. So I want to stop here and give you a sense of where we are now. We start with, on the right-hand side, with a variety of distinct tauopathies that involve different parts of brain and have different regions of involvement. But what we have built up as a model, as a way of understanding, is that a single protein can adopt multiple distinct structures. These can assemble in different ways, and these assemblies create distinct pathologies within the cells of the brain that dictate how they progress, what parts of the brain are involved, and so forth. And so we're excited because we feel like we're beginning to complete the loop, to understand at a mechanistic level how you go from a single protein to a unique clinical syndrome. And the exciting thing is that this model is internally consistent. It's entirely based on the principles originally elucidated that relate to the prion protein. And most importantly, it makes very specific predictions for how we can take care of these patients, how we can intervene, perhaps to stop the initial conformational change, assembly, how we can interfere with transcellular propagation, and additionally, how we might be able to detect these pathological forms in blood or in brain and make a diagnosis before there has been significant neurodegeneration. And I'd like to just stop here and thank all the people who contributed in the lab. This is my current lab. It's big. Everyone, I'm so sorry I didn't get a chance to do justice to more of the work that's going on, but time is limited. And I want to thank my collaborators, especially for this work, Wukash and Bill Seeley, whose work with us really allowed us to move the ball forward. And I'll take questions, and thank you very much for your attention.
Well, thank you, Dr. Diamond, for the wonderful presentation, sharing your insights. For me, the real connection between clinical syndromes and the protein pathology is just striking, and really appreciate it. I'm delighted to introduce our discussants, Dr. Jeffrey Kelly and Dr. Bill Seeley. Dr. Jeff Kelly is the Lita Annenberg Hazen Professor of Chemistry and co-chair of the Department of Molecular Medicine at the Scripps Research Institute. Dr. Kelly's research focuses on the mechanisms of protein misfolding and aggregation in neurodegenerative diseases. He uses this information to develop novel therapeutic strategies. In recognition of his work, Dr. Kelly has received numerous awards, including election to the National Academy of Inventors. His work has also translated to pharmaceutical treatments of degenerative disease, and he is co-founder of Fold RX Pharmaceuticals and Proteostasis Therapeutics. Our second discussant is Dr. Bill Seeley, mentioned several times in Dr. Diamond's talk. Dr. Seeley is the Zander Family Endowed Professor in Neurology. He's also in the Department of Neuropathology at UCSF Memory and Aging Center, and he's the director of the UCSF Degenerative Disease Brain Bank. Dr. Seeley is an expert in the differential diagnosis and treatment of patients with neurodegenerative diseases, and his research focuses on the mechanisms of neuronal susceptibility or resistance to degenerative processes and translating these findings into new treatments. Dr. Seeley was awarded a MacArthur Genius Fellowship for his pioneering research on the mechanism of selective vulnerability of neurons. Bill and Jeff, welcome. Thank you so much for joining us today. I'd like to just remind the audience to continue submitting questions using the Ask a Question button. I think I'll start with the first question, and I think it sort of summarizes several questions that came in. Is there a normal prion function for tau, and how might knowledge of tau structure, such as what you reviewed in your talk, lead to improved diagnosis and treatment, particularly treatment of tauopathies?
We think there is a normal prion structure for tau. Again, I didn't have time to get into this, but we have developed new antibodies that are conformation specific and are very good at pulling out the seeds, the sort of assemblies of tau. And surprisingly, as we started to study diseases, and then started to look at controls, we found that in control brain, there's about 1/10,000th the level of seeding that there is in Alzheimer's, but it's consistently present. What's remarkable is it's only present in the cortex. It's not present in the cerebellum. And it's the amount of this seeding in normal brain is independent of tau levels and age of the patient. So it speaks to the idea that the tau protein at some point in its life cycle is spending a little bit of time as an assembly. That raises the questions of why nature would do this. I have some ideas about this which relate to the utility of prions as carriers of information, a way of building an assembly that might be transmittable either into a new locale in the cell or from one cell to another to transmit some sort of information. We don't know what that would be yet. But it's important, I think, for us all to remember that diseases are just normal processes gone awry, typically, whether it's a growth signal in cancer or, in this case, I would argue, an assembly of tau.
Yeah, I'd be interested in Bill and Jeff's insights into how this might translate into better diagnostics, treatments, particularly translating into pharmaceutical treatments with your work, Jeff, as well. Bill, maybe we could start with you.
Yeah, I mean, there are seemingly endless possibilities, I think, once we start to decode the structural biology of these different tauopathies. You know, from the clinician's point of view, there's still a huge unmet need for being able to see a patient in the office, perform a blood test or a spinal tap, and walk away with a neuropathological diagnosis. We really can't do that for most non-AD forms of dementia. So I think the deep knowledge about the structural biology of different strains of tau is the starting point for being able to move toward that goal. And I'm excited to see what the field is going to do in that regard. I'll let Jeff speak to the sort of therapeutic aspects. Maybe.
I certainly agree. This is really exciting. And I think one of the really interesting questions going forward is how dynamic are these structures? What's the smallest associated piece that can retain the structure and infectivity? We have a lot to learn. But I think Bill and Mark would agree the key in these diseases is diagnosing them early. And so to the extent we can do that and then translate early pathology to more effective treatments, it'll be great.
Thank you. Yeah. The second question is if tau is a prion, is it been shown to be infectious in people?
Do you want Bill or me?
Well, anyone who's willing to take that one.
Well, this is the scary thing, right? Because I think the first thing I would say is I think absent things like brain surgery with tissue transplantation and cannibalism of eating brains, prion protein isn't actually transmissible in people in everyday life. In other words, healthcare workers, caretakers, and so forth are not at risk. But I would say there's no evidence right now that tau is transmissible in a person. And you could, with that knowledge, has been used to say, well, then you shouldn't call it a prion. And I don't really care one way or the other what we call it. Stan Prusner likes to call it a prion, and he's the one who coined the term. So I think when the biology tells us that in almost every other respect, it is infectious, and we can do infectivity experiments the way they do them in the prion field, which is through inoculations of brain. And I actually suspect if you put the wrong structure of tau into an older person's brain, someone who is maybe a little vulnerable, that it could be infectious. But again, that would take some sort of, if you're talking about a risk of a blood transfusion or something like that, I would think that would be very, very low for a variety of reasons. What would you say, Bill?
I don't have a whole lot to add to that. I think it's a harder question to answer with epidemiological tools because some of the tauopathies are quite common in the general population anyway. When someone gets prion disease, it's a 1 in a million event, and so it's easier to find those signals. So yeah, I agree with you that there's no evidence currently. It's a little bit more of a difficult question to study, though.
Thank you. We have a lot of great questions rolling in, so I'm going to apologize in advance for not getting to all of them. But there's a question here that's come up more than once. Does age per se affect tau structure? Are there different findings in younger people, for example, who present with Alzheimer's?
Bill and I'll take a quick, we just haven't looked. I'm very interested in this. We haven't yet applied our methods to a broad enough spectrum. I think the general, I can say, like I said earlier, that there are seeds in young people, and we're still trying to determine what the conformations are. In other words, could they be the precursors of Alzheimer's or something, but we just don't know enough yet. Bill?
Yeah, I think that's fair. I think the cryo-EM literature has also tried to look at this by comparing young and old tau from young and old Alzheimer patient brains and inherited and non-inherited. And the story you might glean if looking at that literature would be that the structures are similar, but for all the reasons that Marc pointed out in his talk, I think that that's just a starting point, and we have to sort of look at the question in multiple different ways using multiple methods to really feel sure. There are a lot of reasons to think that they might be different in terms of the way the patients behave clinically, anatomically, et cetera. So I think there are lots of reasons to keep studying this.
Yeah, I would agree. I mean, I just wanted to highlight, as we do these alanine scans and we're sort of probing the role of the entire molecule in building the structures, it's clear the core is just a very small part of. There's a lot of involvement outside the core, is one of the points I wanted to make, and we can't see that with cryo. you just can't get outside that core.
Thank you. Well, we have time for one more question. Actually, several questions came in about Down syndrome. So I'll just, is anything known about the tau strains that form in Down syndrome? How might they compare to AD tau strains from the general population?
I have no knowledge of that yet. We're hoping to bring out a generation of biosensors by early next year that could be used by a lot of people and would allow a kind of coarse grain discrimination of the different tauopathies. Without, it's sort of like using SNPs instead of whole genome sequencing to genotype people. And I think that's coming. So please stay tuned, and I really hope that we can get, with the new, if we can create a multiplex biosensor system, which I think we're very close to doing, it would allow us to process dozens or hundreds of brains a week, hundreds of brains. We could actually exceed, I'm pretty sure we could go through the entire world's population of the brain banks in months if we wanted to, once this is ready. But it's going to take a little time. It's not quite ready.
Well, clearly this is the beginning of a whole new way of thinking about dementing illnesses, and we really are grateful for this discussion. So I'll just, say in closing that, what I got out of this talk is, as a clinician, there is a very compelling case for classification and taxonomy that goes right down to the protein level. And that's very gratifying and very hopeful. And having Jeff and Bill on really is a great sort of reminder that when we think of it that way, there's hope for treatment, especially if there's early diagnosis. So this really does link to something very, very much on everyone's mind about treatment. So really appreciate that. So I wanted to wrap up by just saying that I wanted to thank Drs. Diamond, Kelly, and Seeley for joining us and really sharing your insights. This is really a great time to reflect on this important topic. And I wanted to thank the audience for joining us and to invite all of you to join the next lecture featuring Dr. Li Wei Tsai on December 7th at 10 a.m. Central Time. And more information will be coming out. I wanted to apologize again for not fielding all the questions. There were some great questions in the chat room, but please feel free to send those questions on to the Oskar Fischer organizers, and we will try to address them at a future time. Thank you very much for everyone's attention.